Hi, I'm RP Jaiswal, and today we are going to discuss about clinical enzymology. So, clinical, which is concerned with the abnormal condition, disease condition, and enzymology means the study of enzymes. So, through the study of the enzymes, we can diagnose some diseases. Some enzymes are disease specific that we are going to learn here. What are the diagnostics significance of the enzymes? First, the enzymes can act as a diagnostic marker of underlying diseases, which means that enzymes are very specific to the diseases. Enzymes can act as a marker, a diagnostic marker for particular disease. So we'll know about what are the enzymes related to specific diseases. Second one is the enzymes can also act as a reagents for various biochemical estimation and detections. So while doing some experiment, while performing some test, we can use enzyme as a reagents, and this will help in the estimation of some biomolecules in the given sample, and we can be able to determine its level in the given sample. So there are two diagnostic significance of the enzymes. Enzymes here we consider as a diagnostic marker, and secondly, enzymes here can be used as a reagent also for detection of the some biomolecules in the given sample. Enzymes as a diagnostic markers. Let's study about this, that how we can consider the enzymes as a diagnostic marker. Here there are functional plasma enzymes, which is derived from the plasma. So they are called as a plasma derived enzymes or functional plasma enzymes. Certain enzymes like proenzymes, their substrates are present at all times in the circulation of normal individual and perform physiologic function in the blood. So what does it mean? That the plasma enzymes are those enzymes which are present all the times in the blood circulation, and their role is inside the blood only. So it has a physiological role in the blood. Example of this plasma derived enzymes are lipoprotein lipase, pseudocholinesterase, proenzymes of blood coagulation and blood clot dissolution. These are the enzymes we have known. These are always present in the blood. Blood clotting factors, for some of them play a role as a proenzymes. What are proenzymes? They are basically inactive form of the enzymes. They are activated by some activators, and they have a role inside blood. So because of this, they are considered, or they are called as a functional plasma enzymes. Means their function is inside blood plasma. The majority of these enzymes are synthesized in and secreted by the liver. So liver is the one of the largest organ of our body, largest gland we can say, and this largest gland of human body synthesizes basically the plasma derived enzymes. Second one, which is not plasma derived enzymes, they are actually cell derived enzymes, non-functional plasma enzymes. So their function is not inside the plasma actually. Their function is, instead of the plasma, their function is inside the cell, because they are synthesized in the cell and the function is within the cell. Plasma also contains numerous other enzymes that perform no known physiological function in blood. So their function is within the cell, not inside the blood. What are the examples? So these non-functional plasma enzymes, basically the examples are the metabolic enzymes, like the enzymes which play very important role in metabolism, the biochemical reactions, carbohydrate metabolism, lipid metabolism, protein metabolism, nucleic acid metabolism. So in different metabolism, there are a set of enzymes are there, and their role is inside the cell. There may be the catalytic role. Yes, enzymes has a catalytic role, means they may have a anabolic role, or the breakdown like effect like role. And these enzymes arise from the normal destruction of the body cells, like erythrocytes, leukocytes, and other body cells. These cells, these body cells, these blood cells or body cells, when they are destroyed, then after that their enzymes leak out into the blood, and because of that we can be able to determine the level of those enzymes in the blood. This is the diagram which is showing the non-functional plasma enzymes or cell-derived enzymes, how the cell-derived enzymes, how it appears in the blood. Here in the normal condition, the A indicate normal cell turnover. There are some cells which are normally destroyed after completing its lifespan. Each and every cell have a lifespan. So when it completes its lifespan, then after that the enzymes of the cell leak out and mix into the blood. The next B1 is the condition where cell necrosis takes place in abnormal condition or disease condition or traumatic condition, injury condition. So when there is a abnormal cell injury, there is a excess breakdown of the cells, and because of that there will be the excess leak out of the enzymes, cell enzymes, into the blood plasma. So at normal cell turnover time or condition, we have a small amount of cell derived enzymes in the blood, but the condition where there is a disease condition, trauma condition, so where there is excess cell destruction, at that condition what happens, the plasma contains more cell derived enzymes. So increased plasma level of intracellular enzymes indicate cell damage. It is due to cell damage. So tissue damage or necrosis increase several non-functional plasma enzymes. Now, possible mechanism responsible for increased serum levels. So what are the possible mechanism where the serum level of the enzymes is increased? So increased condition is due to increased release. So at what condition the enzymes of the cell releases more and more? So one is the necrosis of the cell. Second is the increased permeability of the cell without gross cellular damage, means the permeability, the cell membrane, which stop the movement of the molecules from inside to outside. There is some specific permeability, the movement. But the condition is when the permeability increases without gross cellular damage, means cell is not damaged, but there is a increase in the permeability of the cell. So at that condition the enzymes leak out into the blood, so at a higher rate. Next is the increased production of enzyme within the cell, resulting in increase in serum by overflow. Here the enzyme production might be increased, the demand of enzyme, the requirement of the enzyme is increased, so the production is also increased. Because of that, the serum may contain higher amount of the particular enzymes. So next, last one is the increase in tissue source of enzyme as in malignancy. So now the tissue source, the cell is the source of the enzyme. If the cellular population increases, tissue tissue becomes larger than the abnormal in size because of the increase in the cellular population which occurs during the malignancy. So in that condition also we get increased amount of enzyme in the serum. Impaired disposition, sorry. Here increased level in obstructive jaundice and increased level in renal failure. So here obstructive jaundice and the renal failure is one of the abnormal condition, and in this abnormal condition also we can see that there is a impaired disposition, means impaired increase in the level of those enzymes in this particular condition, and there is an increase in the level of the enzymes in the blood. Decrease the serum levels. Okay, sometimes it happens that the enzymes in the serum may decrease. So what condition can create this decreased serum level? So decreased formation, which may be genetic or acquired. So if the enzymes is not synthesized in the cell, so obviously there will be this decrease in the enzyme level in the serum, and it can be the genetic or it can be the acquired. Second one is the enzyme inhibition. Enzymes might be present in the serum. The enzyme might be synthesized in the normal rate, but there may be some inhibitor present in the enzyme, inhibitor present in the blood or cells. Because of that, the enzyme determination process is inhibited. Enzymes become inactive. So because of that, we may not be able to determine the level of that particular enzymes, though the enzymes might be present in the sample. Next is the lack of the cofactor. Cofactor is the something like it's a non-proteinous part of the enzyme, and they are very essential for the activity of the enzymes. So if there is no cofactor, so enzyme activity obviously it will stop. So because these are the three condition where we might feel the decrease in the serum level of the enzymes. When we measure the enzymes in the serum, so there are some units are there. In that particular unit we have to express the enzymes. So there are two units basically considered. One is the international unit, which is defined as the activity of the enzyme which transform 1 micromole of the substrate into product per minute per liter of the sample under optimal condition at a defined temperature, and it is expressed as IU per liter, means in 1 minute. Okay, how much product is produced from, okay, in 1 minute, 1 micromole of substrate is converted to product. Okay, so how much enzyme is required to convert 1 micromole of the substrate into the product? Okay, so that is the one international unit. Next is cattle. So cattle is also the one of the unit for the enzymes, and here 1 cattle can be defined as the amount of the the enzymes required to transform 1 mole of substrate per second. Okay, 1 mole of substrate per second into the product. So if we have 1 mole of substrate, and if we convert them into the product in in 1 second, So how much enzyme is required? So that is considered as a 1 cattle, and it is abbreviated as a cat or K, small k. Here there is a the relationship between international unit and cattle. 60 international unit is equal to millicattle, or 1 nanocattle is equal to 0.06 international unit. What are the clinical significance of the enzyme estimation? So single or serial assay of serum activity of a selected enzyme. Here we can do this assay means determination of the enzyme. Okay, and this might be helpful in the disease diagnosis. Okay, like it help in the making the diagnosis as well as differential diagnosis or early detection of the disease. So this is very important. Enzyme study is very important, that it helps you in the diagnosis process. It also sometimes helps in ascertaining the progress of the disease. That like the progress means if the person is diagnosed and now being treated. So if you do the enzyme study, and it will help that the treatment is very effective, and it is progressing. Sometimes there may be the condition like the disease progress, like the disease is not curing, it is it is this disorder, means the disease is it is uh, again becoming more severe and severe. Helps next is helps in the ascertaining the response to drug in disease. Like if there is a condition like a person is taking a drugs, so in response to the drugs there may be some enzymes. Okay, and those enzymes can indicate the drugs response, and also help in ascertaining the time course of the disease, like at what time the disease takes place, or the at what time the person becomes sick. So that time of course of disease can be determined by determination of the enzyme. Like it happens like when we have a myocardial infarction, so at what time the attack was taken. So by determining some set of the enzymes, it will give you some idea, the prediction that the heart attack took place at particular time moment. Now summary is that there are some the enzymes as a diagnostic marker. We can see here are a set of enzymes, and they're useful in the particular abnormal condition. Aspartate aminotransferase is increased in the myocardial infarction, sometimes liver disease, especially in the liver cell damage. The aspartate aminotransferase is also known as serum glutamate oxaloacetate transaminase. Next one is the alanine aminotransferase, also known as serum glutamate pyruvate transaminase. So these enzymes are also useful in the diagnosis of liver disease. Again, alkaline phosphatase, which helps in the diagnosis of liver disease where there is a basically biliary obstruction. And the second one is the osteoblastic bone disease like rickets. And acid phosphatase where there is a prostatic carcinoma, and in this condition this particular enzyme is used as a diagnostic marker. Gamma glutamyl transferase, liver disorder like liver cirrhosis, alcoholism. This condition, this enzyme is very important. Now creatine kinase is important when there is a myocardial infarction or skeletal muscle disease. Next is lactate dehydrogenase, which is used as a marker when there is a myocardial infarction, other disease, liver disease, some blood disease. And the next is alpha amylase. Alpha amylase is basically useful for the diagnosis of acute pancreatitis. Thank you. So hope this lecture is useful to study a brief about clinical enzymology. Thank you.